Hi everyone, today we're looking at Chapter 6 from our textbook, Clinical Serology and Immunology. We're looking at Chapter 6, Cytokines. The first slide in the deck really is just for you to get a sense of the kind of the size and scope of the cytokine network that the immune response is influenced by. If you were so inclined, you could take a look at this slide and follow one cytokine on all its paths. So if we look at the cytokine interleukin-1, IL-1, and see, take a look and see where it keeps showing up. So we can see, for example, that it's released from T cells, and it has an impact on macrophages. Gets released, and it affects neutrophils. Gets released, and it affects basophils. Let's see, it gets released, there it is again, and affects B cells. Then what happens? So all of those cells, macrophages, neutrophils, basophils, B cells, they all have a receptor on their cell for this cytokine, interleukin-1. If they don't have a receptor, because again, we're going to see in a few minutes that these are membrane-bound or soluble cytokines, that if they don't have a receptor, they're not going to be able to do anything with the presence of interleukin-1, for example. So we just saw how the T cell releases interleukin-1 and where it goes. Now let's see what happens. So the T cell releases interleukin-1. It affects a macrophage. Oh, and look what happens. Macrophages also release interleukin-1s and they affect B cells. So B cells are affected by interleukin-1 from T cells. Oops, this one's backwards. There we go. Here we go. From T cells and from macrophages. Similarly, let's see, interleukin-1 is released from macrophages and affects basophils. Why? Basophils, B cells, macrophages, they can take the interleukins from a number of different places. B cells can release interleukin-1 and that affects macrophages. They have the binding site, they have the receptor for interleukin-1, so it's where is it going to come from? And so we're going to see this really comprehensive system and how different cells can release the same cytokine, or they can be the recipient of multiple cytokines. So, for example, macrophages, T cells can release all of these interleukins and interferons, and macrophages have the capacity to have a receptor for all of those. Let's see if we looked also, they have TNFs, different interleukins here. So there's really a broad range of receptors on macrophages, which kind of makes sense as a antigen presenting cell and a big player in the immune system. So, as I said, I certainly don't want you to memorize this. I just want you to take a look at it and look at the scope of it, and it may help give you a sense of kind of the process as we look along here. So we've talked about cytokines to some extent before in a number of different contexts. We've talked about it relative to chemotaxis and it's the message that it calls for a cell to move. We've talked about it in reference to hematopoiesis when we looked at cell maturation. We talked about it briefly when we looked at the development of cytotoxic T cells or the development of T helper cells from naive T cells. So what do we know at this point? Taking all of that together, what do we know? We know that cytokines are small, soluble, low molecular weight proteins that are important in regulating both the innate and acquired immune system. We know that. We're going to see at some point that some of them can be membrane bound as well. We have also seen that they play an active role in the hematopoietic system. Those are non-immune cytokines. Those are hematopoietic growth factor or colony stimulating factor. And what those both, they mean basically the same thing. different names, same cytokines. And what do we mean by that? That cytokines, non-immunologic cytokines, can affect the differentiation and maturation of hematopoietic cells that are developing in the bone marrow. We know that beyond this regulation, or we can further talk about that regulation in a sense of kind of the functionality of cytokines. We know that when cytokines get induced by a specific stimuli, maybe it could be a bacteria or a virus or some other kind of a pathogen, they act as a chemical messenger between cells, kind of like a telephone line between cells. And those chemical messengers participate in a number of different immune responses and immune functions. So what kinds of things can, once those chemical messengers get released from a cell, how can they exert effects on other cells? What do they cause to have happen? We've talked at some point about regulation, some of the regulation functions of cytokines. They can regulate the intensity and duration of immune response. They have that capacity to suppress an immune response if it's going on too long. They have the capacity to stimulate an immune response. We've seen how they can activate proliferation, whether it's within the bone marrow or within let's say, the adaptive immune system when we saw the proliferation of T helper cells. That was under the action of a cytokine. We've seen how they can influence cell development and differentiation. And we've seen how they can perhaps act to trigger the production of and secretion of antibodies. Or they can even influence the production or secretion of another cytokine. So that regulation and processing of our chemical messengers, our cytokines, those are the effects that cytokines can have on other cells. What else can it do that's both familiar to us and we're going to look at in a little more detail? We're going to see today how it influences inflammation and the processes of the innate immune system. We're going to look at how it recruits, and we've seen how cytokines, specifically chemokines, can recruit and cause other cells to migrate to an area where they're needed. We are going to see how it can really influence other actions today. We haven't spent a lot of time on this yet, but we'll spend more time on it today, and really that there are some cytokines that will influence, for example, enhancing of an acute phase reactant within the innate immune system. As I said, just as a reminder, cytokines bind to very specific receptors on target cells. If a cell does not have that specific receptor, the action of the cytokine does not take place. Let's see, let's move on. I think that will be good. In general, we know there are well over 200 cytokines in the human body. Originally, cytokines were named after the cells they were isolated from. So cytokines were secreted from lymphocytes, were lymphokines, from monocytes, in macrophages were called monokines, and those cytokines released from leukocytes that act on other leukocytes were called interleukins. Makes sense, right? I think we're good. Now, though, I think we've discovered that it's far better. Our original thought about some of the cytokines were, we thought that they were functional based on where they were produced, and that's truthfully not the answer at all. So we're really looking, when we classify cytokines, we classify them and group them by family, by the family name. So we group them into 1, 2, 3, 4, I think it's 6 different categories or families. The TNF family, the tumor necrosis factor family, interferon family, chemokine, transforming growth factor family, colony stimulating factors. You may see them as the hematopoietic growth factors. In your book, I think, yeah, they call it hematopoietic growth factors in the book, but they're CSF cytokines within that. And then the interleukins, the IL, and they're numbered. You may also see some family names that look like this. So those are just a slightly different version that I've seen in other texts that don't really include the culinary. Oh no, well it does. It doesn't call it the CSF factors. They call it the hematopoietic family. So they're just a little bit different. But this is the terminology we're going to be using today, and I think this is probably the most current terminology that we would be using. What more is there to know? Well, there's an awful lot. But let's start with the actions of cytokines. Cytokines don't act alone. Cytokines just don't release out and cause something to happen. We know that they have to have a receptor in another cell, right? In order to work. So they work, cytokines are chemicals that work in coordination with other cells, and when they're released by cells, an effect or interaction or communication, whatever that signal is that gets sent, has an effect on whatever cell is the recipient. So we have one cell acting as the speaker and is sending out that chemical signal, that chemical message, and another cell is going to pick up that message and hear it, quote unquote, hear it, and have an action take place. So we're going to look at these three terms in a few minutes. I just want to make sure we're aware of them. So the reality is that cytokines, when let's say there's a cell that is releasing a cytokine, that cytokine gets released, gets produced and released. That cytokine can actually come back and affect the same cell and cause an effect that way, or those cytokines can get released and affect another cell, or those cytokines can get released and go into the bloodstream and impact cells far, far away. And that's what these three terms are going to be. And we'll go over those in just a few minutes. The release of cytokines from a cell causes what we call the cytokine cascade effect in order to stimulate an immune response. So we have that first action of a cell releasing a cytokine, right? And finding another cell with a receptor on it. That's step one. If you think of this little domino as being step one, that's fine. So then maybe this cell, here's the cell, let's pretend the dominoes are the cells. So this first cell is the one that is maybe been exposed to a pathogen or has been triggered by something. Could be a flagella, could be a number of things. But this cell is triggered to release cytokines. The cytokines are secreted and produced, secreted, and released and act on this cell. This cell then has an action. Maybe it is going to proliferate. Maybe it is to differentiate. We don't know what it is. But it's going to have an action, and the action of that is going to influence another cell and cause another domino to fall. And then this one, something else happens. Sometimes these are the same cell type, sometimes they're different. But remember, this same cytokine that gets released from this cell maybe doesn't just affect this one. Maybe it affects this one as well and comes in from a different angle. So you're going to remember that the cytokine cascade is really like dominoes falling. They line up. The release of one cytokine can trigger and activate another cell or some other cells to produce other cytokines or to produce an action. We'll see an example in just a few minutes, but remember, the same cytokine can be released by really one or several types of cells, and the human body was pretty well designed and has manifested over time to have a lot of systems that make it so that it's kind of foolproof. And the human body has some redundancy built in it to it, and that's why one cytokine can really affect a couple of different cells, or different cytokines can activate the same pathway or genes to cause the same action. So we have that redundancy built into our cytokine system. And redundancy really just is two mechanisms or more to get the same thing done. And we're going to see that in a few minutes. So the process of producing and releasing cytokines in the immune response begins with the stimulation of a cell of the immune system in response to the identification of a pathogen. So usually secreted by, T cells, monocytes, macrophages, B cells, natural killer cells. Cytokines also can be released from other things. They can be released from what's called a Schwann cell, Schwann, Schwann cells. Those are in the central nervous system. Epithelial cells, endothelial cells, things like fibroblasts. But really, once they're secreted, cytokines bind to the receptors on another cell, and those are going to allow the recipient to receive the message. The originating cell sends the message, the target cell receives the message via a receptor on its cell surface. This receipt of the message, the chemical message, allows the recipient cell to then respond with an action. That's that cascade, the cytokine cascade that I spoke about. So for example, a macrophage phagocytizes an antigen. They present the antigen peptide, and cytokines are released, which bind to receptors on B and T cells. Those then stimulate specific immune responses by B and T cells, and also some non-specific immune responses, such as inflammation, all from that same initial pathogen identification. So we're going to break down the actions of cytokines really into smaller pieces over the next couple of minutes. Again, just, this is a great opportunity to just see and recall the original cell sending the message and the recipient cell receiving the message. So we could, rather than looking at a large piece, let's break the action of cytokines that I just walked through for you into some really small pieces. I gave you the example of a macrophage being stimulated after having ingested a pathogen, presented it. Cytokines are released. They bind to receptors on a T or a B cell, and adaptive immunity begins. Let's look at the actions of cytokines in really, kind of broken down into small pieces here. I think this will help. The illustration on the right is, honestly, it's about as good as I've seen in breaking it down for people. And I think it's going to help. So, a cytokine producing cell out here is stimulated. A cytokine producing, right here. A cytokine producing cell is stimulated by, again, an antigen of some sort. It could be anything. It's stimulated. Maybe it's we talked about how there can be components in bacterial cell walls, or the presence of a flagella on a bacteria. Something induced a stimulus here that stimulated this cytokine producing cell. Once the cytokine producing cell has been stimulated, the genes within that cell, the genes that are going to influence cytokine production, is activated, and the results follow that the cell then produces and secretes cytokines. So cytokines are produced within the cell and they're released, and that's in direct response to this original, let's say, antigenic stimulus. So the cytokines are released. The cytokines that were released then act only on the cells that have a receptor for the cytokine. If the cytokine receptor is present, then the cytokine binds to the receptor, and that triggers additional gene activation of the target cell. We call this the target cell, the one that's waiting for the action of cytokines to make a move, right? So it's going to trigger the activation of a gene within the target cell, and that's going to result in some kind of a biological effect. Maybe it's proliferation of the cell. Maybe it's further differentiation of the cell. Maybe it's cell death. Maybe it's apoptosis. We don't know what it's going to be. What else do we know about this? I think that's it for now. Let's look at this and break it down just into a little bit more specifics of what I just walked you through, just so you could see it in writing. I wanted you to first see it and hear it, and now I want you just to have the breakdown here. So again, the stimulation was induced, and we say, for example, that could be a bacteria that will trigger an immune response. And again, maybe it's bacterial waste products or a flagella, the presence of a flagella. All sorts of things can trigger and act as a stimulus. But there's got to be something that stimulates this cytokine producing cell. Often we see it in the immune system that it's presentation of an antigen. It's been the processing by an MHC class and presenting an antigen. That is a inducing stimulus. So it could be anything. It activates the cell, the cytokine producing cell, to synthesize and release cytokines. And we frequently think of this cytokine producing cell as being a lymphocyte. We saw that an awful lot when we were looking at antigen presentation. If we remember, what kinds of T cells are the recipients of antigen presentation and the cytokines that get released as a result. They were T helper cells, right? So they were lymphocytes. This is frequently a lymphocyte and a T helper cell, a naive T helper cell, right? So they release cytokines. This is your antigen. This is some sort of an antigen presenting cell. It was a stimulus. This could have been a T helper cell. It could have been a macrophage. It could have been anything. But again, we think of this often as a lymphocyte. Cytokines get released. They find a receptor on a target cell. These cytokines could have been one of those interleukins. It could have been a TNF, a chemokine, whatever. And those are going to all have their own effect on this receptor cell. It's going to trigger that receptor cell to do something specific. They bind to the targets on the receptor cell. And remember, if, for example, I gave you the example, if interleukin 4 is released here from the cytokine producing cell, the target cell has to have a receptor that's an interleukin 4 receptor. And then the biological effects: cell death, suppression of immune response, trigger of other cells, proliferation, cell differentiation, whatever the case may be. Let's see. So in this slide, I know where you're saying, are we looking at the same thing again? Yes. So I know in this we just broke down how cytokines bind to the receptors of the target cells. But now we're going to look at really the actions of cytokines binding to specific receptors on a target cell and how they trigger a genetic response. If a cytokine gets released, the target can be any number of different kinds of cells. And it's relative to the originating cell. So let's say, for example, this cell right here is our cytokine producing cell. And this cytokine producing cell releases cytokines And look, this cell happens to have a receptor for the same cytokine on its surface. We call this action of cytokine release and triggering of the target cell an autocrine reaction. In this case, the cytokine producing cell and the target cell are one and the same. So that autocrine reaction is just referring to a cell that can release cytokines that influence itself to do other things. The next kind of action that we're going to take a look at is it's called a paracrine action. And all that's saying is, here's again the cytokine producing cell. This cell and this cell are the same. And it releases the cytokines. And this target cell is just nearby. It's close by, but a target cell nearby has the receptor of the target cell. So in this case, a paracrine action is just referring to a cytokine producing cell that releases cytokines, and they act on a cell, they bind to and act on a cell nearby. That's paracrine. And finally, we have a case where it's called an endocrine action, where the cytokine producing cell releases cytokines, but those cytokines go into the blood vessels and they go into the circulation. And they get released through the circulation, and they find a receptor on a cell far away and distant. We call that action an endocrine action. Let's see what else. So the effects of the genetic activation of target cells by cytokines, what can they do? They can regulate the gene activation and expression. We saw how they can affect growth and cell differentiation. So those are some of the actions of these cytokines that can take place. But they're exactly what we expect to see and what we've talked about. And because some people like a nice visual like this of what the autocrine, paracrine, and endocrine actions are. Some people like to just see it in this fashion. So here are the terms we just discussed and their definitions. It's important to remember that, this is a simplistic view of the work of cytokines. And in fact, cytokines really they don't go to work alone. They work as a group. They work in conjunction with other cytokines. They work they work together to either stimulate or suppress actions of target cells. If you go back to that first slide in the deck today, we see those families of cytokines being produced and influencing the actions on cells all throughout the immune system, and they're from cells, different kinds of producing cells. There are different kinds of cytokine producing cells. So, when we look at it this way, it's like, oh, it's pretty simple, but it's really not. It's a very complex process. But we're going to continue to keep breaking it down into small pieces for us today, and we'll work through it until we get to something complex at the end. Before we leave the effects of cytokines on the target cells, let's look at, I think this is in the book, figure 6-1 on page 78. This gives an overview of the cytokine story that we've been telling. In this illustration, the antigen presenting cell here and that's of course what? The cytokine producing cell. The antigen presenting cell releases interleukin-1 beta. And so if we look here, interleukin-1 beta, and it has an effect on itself. It feeds back to itself and causes more production. It may cause more production. It may stimulate other actions as well. So that interleukin-1 acts on itself in an autocrine action. It affects cells nearby, like neutrophils, and in a paracrine action. Or it actually also can affect nerve cells in a paracrine action. What else can it do? That same interleukin-1 can go into the vascular. This was a really bad picture of a blood vessel. It took me a few minutes to figure it out. I labeled it because I was like, what is that supposed to be? Oh, it's supposed to be a blood vessel. Helps when you read the book. But if it releases from the antigen presenting cell, those same interleukins get released from the antigen presenting cell and go into the blood vessels. They travel through the vasculature and go far, far away, and it reacts with cells of the liver, for example. It had a couple of effects. Interleukin-1 can affect neurons in the brain, and it can also reach the liver to stimulate some acute phase reactants release, for example. This action in the brain, it can stimulate a fever, believe it or not. Interleukin-1 affecting cells in the brain can stimulate a fever in the hypothalamus. The hypothalamus acts like a thermostat in our body, and when it's under the action of cytokines of the immune system, it turns the heat up a little, and basically bacteria just don't live and thrive really well in hot bodies, and we'll see the bacteria, the pathogens die, whether it's a bacteria, maybe even a virus. So this is what we've been talking about. So there is the antigen presentation to the T helper cells. So the interleukin-1 also affects this process in that it's going to cause the T helper cell to further differentiate from this naive T helper cell to whether it becomes a cytotoxic T cell, or it's going to trigger the release of what was I going to say? Immunoglobulins. So yeah, a lot of different actions from the actions of this one antigen presenting cell that had processed and presented an antigen. And the release of the cytokine, the same cytokine, had an action on a number of cells. Let's see. Cytokines, let's start again. We thought that once, the theories of immunology really thought that cytokines only acted on cells of the immune system, but we know that that's different today. We've just seen it in action. We saw the endocrine system, for example, where the endocrine action, I should say, where interleukin-2, can't believe I've already forgotten what it was, interleukin-1 had an effect on neural cells as well as the liver. So those are clearly not cells of the immune system. We're going to see another example in just a few minutes. But when we look at each of those families of cytokines in more detail, we'll see how those actions can affect non-immune and immune cells. But for now, we're going to remind ourselves that cytokines don't just interact with cells in the autocrine, paracrine, and endocrine actions, but they also have a different kind of personality. They functionally have differences in their actions, in their functionalities. And so we say that functionally, cytokines can exhibit a number of different attributes of how they work, their functionality, as it were. So the autocrine, paracrine, and endocrine really refers to kind of the actions of the cytokine. What's it going to act on? What cells is it going to affect? The functionality that we're going to talk about now really talks about how the cytokines that get released can have an effect on a number of different cells in different ways, and how they work as a team. Remember, it's not on this slide, but cells work in a team. Cytokines work in a team, right? They work as a group. So as long as we remember that, we're gonna be able to move forward. This functionality that we're going to talk about, pleiotropy, redundancy, synergy, antagonism, and cascade induction, those are the functions of cytokines that we're going to talk about. And these are all going to discuss really about how cytokines regulate the activities of cells in a very coordinated way. So let's take a look. Let's look at it. It'll make a little more sense as we go forward. So functionally, cytokines can act, I can already see, I have a typo here. This should be P-L-E-I-T-R-O-P-Y. I don't know how I missed an I in two places, but I did. Pleiotropy. Pleiotropy is the functionality that says that one cytokine can have a different effect on different target cells. Going back to our very first slide in the deck, I gave you the example of interleukin-1 that was released from T cells. And if you want to go take a second and go back to that slide in your notes, you can see that interleukin-1 was released from T cells, and it had a number of different effects. It had different target cells, and those target cells also released interleukin-1 in different ways. Remember, as long as there's a receptor there for that cytokine, it is going to cause an action. So cytokine is a single, pleiotropy is a single cytokine that has a different effect on different target cells. This, ignore this. Here's an example of pleiotropy. So we have an activated T helper cell that releases interleukin-4. Interleukin-4 acts on target B cells, so there'd be a receptor here for interleukin-4. And what impact does interleukin-4 have on B cells? Well, it provides activation, proliferation, and differentiation of B cells. That same interleukin-4 acts on thymocytes, and it causes proliferation of thymocytes. And that same cytokine, interleukin-4, when it binds to, I can't even see what it says, mast cell, must be a mast cell. When interleukin-4 binds to a mast cell, it causes proliferation of mast cells. So one cytokine has different effects on different target cells. Different effects, different target cells. Does that make sense? That's pleiotropy, and I will correct this in the slide deck I'm going to throw on the portal. Sorry about that. I think you'll find it's spelled right everywhere, except right there. So beyond pleiotropy, what other functions, what other properties of cytokines are going to be there? They're going to help us view the way they regulate the activities of cells, kind of in that team building way, that coordinated way. Well, there are three other ways. In the case of redundancy, actually this should be 4. Yep, there'll be 4. But in the case of redundancy really is just referring to the fact that different cytokines can activate the same pathways. And this creates kind of that backup system that is fantastic for a host. So, for example, in this slide right here, we see an activated T cell. And an activated T cell can release interleukin-2, interleukin-4, and interleukin-5. And in all 3 cases, they result in the proliferation of B cells. So if you had some cells that had only a receptor for interleukin-5, then it would proliferate still. The cell would still proliferate. So it's kind of a backup system that different cytokines can activate the same pathways and have the same effects on the target cell. Does that make sense? Okay. Synergy is one where multiple cytokines work together in a synergistic way. It's exactly what it sounds like. So interleukin-4 on its own is able to change when it acts on a B cell. it can actually trigger that B cell to stop producing IgM and to switch over to produce IgE. That's the action of interleukin 4, among other things. But interleukin 4 and interleukin 5 together enhance the trigger to make B cells class switch to produce IgE even stronger. And it makes it so that they produce something other than, it makes them produce IgE instead of generally IgM. So this is, again, synergy. Two working together are better than one alone, is really what that's saying, right? Synergy. And antagonism is exactly what you'd think. So antagonism really means the action of one cytokine can influence or counteract the action of another. In this case, that same interleukin 4 that we just saw that will cause a class switch when it finds its target receptor on a B cell, it can, remember we said that it can induce a class switch from IgM to IgE. Well, if we have interferon gamma released as well from that same activated T helper cell, it actually blocks the receptor on the B cell, and by virtue of that, it blocks the switch from IgM to IgE. This is a really helpful regulatory response, right? So this is great for stimulation. This is great for regulation. And that's the example that we see of antagonism versus synergy. And again, redundancy is just making a redundant system. It's a backup plan. It's a backup method. It's a little bit of a funky graphic that's in your book. It actually, once you see it, it puts it in a different perspective, whether you like this or not. I just want to walk through it. Sometimes people like it and they can see something in a different way. So this illustration really was showing you the difference between synergy and antagonism. So if we have, for example, there are two cytokines that we're looking at. Really three, but we're looking at cytokine A, cytokine B, and cytokine C. Doesn't matter what they are. Cytokine A alone is, we're looking at cytokine B, and we're looking at the response of cytokine B. So when cytokine B is working on its own in very low concentrations of cytokine A, it's doing okay, but it's not having a great deal of effect on the target cell response. It's not finding a lot of targets, and it's not making the whatever it is, proliferation take place or differentiation. It's a very slow process. But as the concentration of target of cytokine A, I apologize. Let me start again because I don't want to confuse you. So this is the concentration of cytokine A. Right down here, it's very low. See how that says it's a 0 concentration? B didn't start at 0. B started at a different, random number. But right now, there's very low concentration of cytokine A that's available. It's not being produced for some reason. And cytokine B is being produced. And it's supposed to have an effect on a target cell. It's supposed to cause a target cell to do something, right? But by itself, it's kind of a slow and steady action on the cell response. But when you start to see that cytokine A is growing in a high enough concentration, once it's in a high enough concentration, it can work in synergy with B, and all of a sudden B, the action and the target cell response, starts to be on a very upward trajectory. So A synergizes with B to cause an increased target cell response relative to B. So B is the one that's causing the action, and A alone would be kind of the secondary one. Conversely, if we have C, even in very high concentration, or if C is a cytokine that's having an effect on a cell response, when it's by itself, it has a very high response to the target. The target cell is overdoing whatever it's supposed to be doing. That when we start to develop some increase in concentrations of cytokine A, they antagonize C, and the action of C on the target cell diminishes in proportion with A, the production of A. So this is really just showing you the synergy when A and B work together and the antagonism when A and C work together. Whether you like this or not, I don't know. I did want to go through it with you in case you took a look at it in the book. I prefer this model. I think to me this is a lot more intuitive. This I think is a little bit tougher, but maybe you like it that way. But I did want to go through it because it was in the book and I didn't want you getting stuck on it. And we talked about the functions. Remember we talked about pleiotropy, redundancy, synergy, antagonism, and there was one more on that list, and it was called cascade induction. Actually, if we go back, there's our list, right? Functionally, cytokines can exhibit these functions, right? So, cytokine cascade induction is where we are now. Again, visualize this as a row of dominoes falling. The first one to fall causes the next to fall, causes the next to fall, and the next. Remember though, one domino can cause two different paths to be activated. You know, you see it all the time if they split and one domino just hits the corner of two different pathways. It starts both pathways at once. And eventually all of the dominoes fall. It almost seems the more you're doing, the faster it gets. You know, beyond that first trigger. The first trigger sometimes seems slow, but once it gets going, those dominoes falling seem to go really quickly, right? That's a cascade in action. So normally, when a secreted cytokine activates T cells, it will cause those target cells with the receptor to secrete other cytokines. This is the domino effect that we see. So when one action of one cytokine on a target cell induces that cell to produce another one or more cytokines, that activates other target cells to produce more cytokines and more biological reactions, cell death, differentiation, proliferation, whatever the biological effect is, that's a cascade induction. That's a cascade cytokine cascade. Normally, so there's normally though not just that the secreted cytokine can activate the target cell to produce the cytokines and that cause that rapid generation, that speeding up of the cascade of the acquired and innate immune systems, but there's usually a feedback loop. And what the feedback loop does is it's a regulation. A positive feedback loop is going to keep calling for more and more of the cytokine cascade, the cytokines to be produced, and it's going to keep going. This sometimes can result in an overproduction of cytokines. This overproduction that, we're just constantly producing more and more cytokines and stimulating more immune cells can result in something we called hypercytokinemia or a cytokine storm. It's just about as bad as you'd think it sounds. That massive overproduction and really the inability to regulate the cytokines through a feedback loop, whether it's positive or negative, results in this overproduction of cytokines and causing a cytokine storm. Let's see what happens in let's look first at the cytokine cascade induction. And we'll talk a little bit more about cytokine storms in just a minute. This is not as clear as I would hope. I'm sorry, but I think we'll get the point. This is the activated T cell that's released a cytokine. It released interferon gamma interferon. That then has an action in that it binds to the receptor cell on a macrophage. So there's that, right? Once the gamma interferon has bound to the receptor on the macrophage, what happens? It triggers genetic activation within the macrophage to do what? Synthesize and release interleukin 12. So the macrophage is triggered by binding of interferon gamma to its cell surface, begins the genetic activation to synthesize and release interleukin 12. Once interleukin 12 then can bind to a receptor on another activated T cell. It actually could loop back to this activated T cell, but in this case we're going linear. So interleukin 12 binds to a receptor on another activated T helper cell. And now now that binding has taken place, that the interleukin 12 has bound to the activated T helper cell, these T helper cells then can release a number of different cytokines. We had a couple of examples of gamma interferon, tumor necrosis factor, and interleukin 2 being released from the T helper cell as a result of starting this cascade induction way back here. The gamma interferon, the tumor necrosis factor, and the interleukin 2 and the other cytokines that get released then bind to targets on, if we had additional cells below this with receptors, and they trigger an ongoing response. So the actions of those just keep going. And this is the cascade that we're talking about. The cascade induction starts where? Right at the top, where activated T cells, as a result of, as I said, any number of different kinds of activators. It could be antigen presentation. It could be the presence of a particular bacteria or something about that bacteria, or a virus. That's what starts the cascade induction. Normally, this is self-regulating. It's not going to go out of control. But if this feedback doesn't stop itself if it doesn't regulate it when things are under control, if the cytokines get produced in too great of a number, we have the possibility of creating what's called a cytokine storm. Again, cytokine storm is just that massive overproduction of cytokines. It has a lot of really negative effects that can happen. Can cause hypotension, fever, edema,